Hello everyone, I'm HEERANYAA Nadaraja and I'm from DBT 4A. Today I'm going to perform on bacteria enumeration. Enumeration of bacteria. The laboratory microbiologist has often to determine the number of bacteria in a given sample as well as having to compare the amount of bacterial growth under various conditions. Enumerations of microorganisms is most important in microbiology, food microbiology, and water microbiology. Knowing the bacterial count in drinking water, fresh milk, butter, buttermilk, and so on can be useful in many aspects of industrial microbiology. Bacteria is so small and numerous, counting them directly can be very difficult. Some of the method it will be used involve diluting the sample to a point at which the number of bacteria has been reduced to a very small numbers. Here we go. We are going to select the variable plate count. What is variable plate count? Variable plate count is also known as the standard plate count. It's the one of the most common method for enumerations of bacteria. The serial dilutions of bacteria will be plated onto an agar plate, and dilution procedure influences overall counting process. The suspension will be spread over the growth of the on the surface of the growth medium and the plates will be incubated uh, for the colonies to be formed. Many bacterium on the solid media results will be formed of a microscopic colony visible to our naked eyes. And the counting plate should have 30 to 300 colonies at least, or we also can be have 25 to 250 colonies. It can be in these two ranges. These are the furthermore explanation about the variable plate count method. Here we are having some briefing about the variable plate count. So basically this is our procedure and this for our samples. We are having water from a drinking fountain, boiled water from a drinking fountain, water from a local river, boil water from the local river. So these are the four materials that we are going to use for this experiment. So this is our procedure. First we will take about 6 dilution tubes. Each will be containing 9 ml of sterile saline, and we will be dilute 1 ml of the sample by withdrawing 1 ml of the sample and dispensing this one sample into the first dilution tube. So basically we will take out this 1 ml from the sample and we will be dispensed into the first tube. Using the same procedure, withdraw 1 ml from the first dilution tube and dispense into the second dilution tube. Subsequently, withdraw 1 ml from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is completed. So, first we will take out around 1 ml of this sample and we will be dispensed into the first tube. Then this 9 ml will become 10 ml. Therefore, we will continue the same step for the other tubes, like taking about 1 ml from this first tube, first dilution tube, and then we will be transferred into the second tube. And then taking another 1 ml of this solution, we will be transferred into the third tube. So this will be continue until the last tube. Then we will be transfer around 1 ml from each of only the last 3 dilution tubes onto the surface of the corresponding agar plate. Incubate the agar plate at 37 degrees Celsius, which is known as optimum temperature, for 48 hours. So it's basically like 24 to 48 hours. And we will be choose a plate that appear to have between 30 to 300 colonies or 25 to 250 colonies. And we'll be count the exact number of the colony on the on that plate. The calculation of the number of CFU, which is colony forming units per milliliters of the original sample, will be like this. This is the formula for to calculate the CFU per ml of the each samples. So we will be counting um, the colonies between 25 to 250 or 30 to 300, and there will be times with the dilution factor of the plate count, which is it will be like 1 over 10, 1 over 1000, like that, but we will be count for the last three only. So it will be like 1 over 1, that 1 over 10,000, 1 over 100,000. It will be like that. So let's proceed with the procedure. Click to the dilation tube. We'll be click the dilation tube with a rack of test tubes to bring them to the table, and then each dilation tube will be contain around 9 ml of sterile saline solution, and then we will be click on the water sample, then we'll be click on the eye drop to withdraw about 1 ml of the sample. So the first sample is the fountain water. So around 1 ml of the sample will be dispensed into the fountain water. Then click next. Again we will click to the eye drop. Okay, we can use the same thing. So here you can see we are taking about 1 ml, 1 ml, 1 ml, and we are transferring it. Then we have to click next. Okay, here is the theory about the dilation tube. The dilution for each of the 6 dilution tubes can be summarized in the image below. So the dilution tube 1 has 1 over 10 dilution with the dilution factor of 10. So the dilution factor for each tubes will be like, for example, if the tube is 2, so it will be 1 out of 100. If the tube is 3, it will be 1 out of 1000. If the tube is 4, it will be 1 out of 10,000. So here it will comes like that. Then next we will be incubating the agar plate into the last 3 broad culture dilutions. Let's click on the agar plate. Then we'll be click on the eyedroppers to transfer about 1 ml of the dilution to the tube number 4 to 1. Then about 1 ml of dilution from tube number 5 to plate 2. Then about 1 ml of the dilution from tube number 6 to plate number 3. Then we're going to click on the pencil to label the agar plate 1, 2, and 3. We're going to label it like 10,000, 100,000, and 1,000. Click next, and then we will be incubate this agar plate into the incubator at 37 degrees Celsius for 48 hours. Let's click next. Now we're going to click on the incubator to see our colonies in the agar plate. So here the plate number 1 is the fountain water. This contain the sample of the fountain water, colonies of the fountain water. Plate number 2 is the boiled fountain water. Plate number 3 is the river water, and plate number 4 is the boiled river water. Let's click number 1, which is the fountain water, to see the colonies on the plate. So let's click 1. Here we go. You can see the colonies for 1/10,000 dilution. The dilution factor for the plates that we are viewing is 10,000. And then this is the dilution factor that we are viewing is 100,000. There are few only. You see here is decreasing. Here will be more. Here it will be less. So it's decreasing. Decrease. Here we can only see there's one colony. Next, let's look for sample number 2, which is boiled fountain water. Here is 10,000 dilution factor for boiled fountain water. Here's 100,000. Here's for 1 million. There's no colony here. And then let's look for sample number 3, which is river water. So 10,000 dilution factor plate. You see here got so many. Will be more than 300 maybe. It's quite less. It's lesser. And then now we are going to look for the colony's growth in the boiled river water sample agar plate. Here we go. This is a dilution factor for 10,000 for boiled river water. This is a dilution factor for the plate for the 100,000. Here is for the dilution factor for 1 million of the boiled river water sample agar plate. That's all. That's all from me. Thank you, sir.